Good afternoon, everyone. Welcome to this SMCB online platform. So we are happy to host today Dr. Snehasis Daschakraborty from IIT Patna. He has done PhD from SN Bose Center for Basic Science, Kolkata in the year of 2014. He worked with Professor Ranjit Biswas there, and then after that he moved to Department of Chemistry and Biochemistry at University of Colorado to work with Professor J.T. Hines for his postdoctoral research. And thereafter, he joined IIT Patna. So he is working at IIT Patna since 2016, December, and he has been recently promoted to associate professor. So he has many awards and recognitions. For example, he got the INSA Medal for Young Scientist. He got CRSI Bronze Medal. He has also received JSPS invitational fellowships to visit Japan. And he has extensively working on the topic which we has chosen today. So he will be talking about jump dynamics, whatever his understanding is to this platform. So without further delay, I request Snehasis to share the screen and start today's talk. Snehasis.
Okay, thank you, Rajiv. I will share.
Yes. Okay. Yes.
Yeah, I think the screen
this is the first list. Yes.
visible, no?
Okay. Yes, it is visible. Please go ahead.
Thank you, Rajiv, for your nice introduction. And I want to thank Professor Bakshi for and also the coordinators of SMCB lecture series, Sumanda, Professor Biswas, and Dr. Rajiv Biswas for giving me the opportunity to talk in this beautiful platform, SMCB. So today I will talk on this topic, unlocking supercooled water, translational jump-diffusion approach, and broader applications, which we are working for the last 7.5 years or so as I joined IIT Patna. So before going to the topic, I will briefly introduce the works we are doing, different other works we are doing in IIT Patna. So we work on the dynamics of supercooled water, adaptation of cell membrane of externophiles, where we try to understand the adaptive strategies of cell membranes of different external organisms like cycrophiles, thermophiles, barophiles, etc., which survive and thrive at very extreme and hostile conditions. We also work on different amphiphiles, like normal surfactants to aroma molecules, and also these different liquid crystal molecules, which are amphiphilic in nature, and how they form these thin layers on water-vapor interface with collaborations with experimental groups. We also worked on nanoparticle cell membrane interactions, and lastly, the lateral dynamics of lipids in cell membrane. So today, I will talk on this topic, dynamics of supercooled water, and I will also touch on this lateral dynamics of lipids of cell membranes. And it will come naturally as an applications of this jump diffusion approach. So, I will introduce the topic, and the first 15 or 20 minutes or so will be for the students who are new in this area. So this is the topic, and you can see this perspective article we have written in 2021, where the methodology and the methodology of this jump dynamics, jump diffusion approach has been summarized. So question is, what is supercooled water for the beginners in this topic? Supercooled water. So before going to supercooled water, let us see the phase diagram of water. You can see here this is the typical phase diagram of water we read starting from BSc days, that you have different phase coexistence line, and this different phase coexistence lines actually separates the phases of water: ice, liquid water, and steam. And as you see, this two phase coexistence line, water and ice, this line, when you cross this from liquid water below certain temperature, that is called freezing or melting temperature, water will freeze into ice. And for atmospheric pressure, 1 atmosphere, this temperature is 0 degrees Celsius. So below 0 degrees Celsius, we think that water will be frozen into ice. But that does not happen, particularly when there are absolutely no impurities in the water, and also cooling rate is significantly fast. So in that case, you can get water in the liquid state below the freezing point, and that water is commonly called as supercooled water. So it can be even more clear if we see this schematic free energy diagram, free energy profile for at these 3 different thermal temperatures. Below above 0 degrees Celsius, 273 Kelvin, you can see that we have 2 minima, and the liquid water is the minimum for the liquid water is much deeper compared to the ice, and therefore we can say thermodynamically liquid water is more stable than ice. But if you go to the 0 degrees Celsius for atmospheric pressure, for example, you can see that a double well and the depth of the well are same, and therefore liquid water can be easily transformed into ice, and also back to liquid water. The overall free energy differences is 0, and therefore we call that is in equilibrium. Liquid water and ice are in equilibrium, thermal equilibrium with each other. So this thing will be reversed.
Hey, Snehasis, I have just a small question. In the phase diagram, PT diagram, where is your supercooled water? That's for students especially. Can you
I'll go into that in detail.
Yeah,
so this is supercooled water is not yet shown in this typical phase
yes,
diagram.
Yeah. So I cannot see that actually. Yeah.
Yes, we cannot see that. I'll go into that in the next slides.
Yeah.
So here what happens is below 0 degrees Celsius, you can see that water is stable at the less stable compared to ice, and therefore if you start from liquid state, if you can give this free energy barrier, it can easily convert into ice and thermodynamically more stable state. So supercooled water is basically metastable, and it requires some activation energy to freeze, and therefore water in supercooled state is slightly difficult to keep, particularly at very low temperature, because as the temperature decreases, this minima will increase. So question is, supercooled water is available in nature? Answer is yes. In altocumulus cloud, you can see, particularly in the hole punch clouds, there are pockets where supercooled waters are available. And this is particularly important because when aircraft passes through it, then there will be a quick nucleations and there will be freezing, and that can harm aircraft. Biologically also, supercooled water, some externophiles, as I said, externophiles, particularly the psychrophiles, which survive and thrive at very low temperature, they have the strategy to keep water in liquid state even below 0 degree, far below 0 degrees Celsius, with the help of many different things. It can be proteins, it can be different osmolytes. So these are available in nature. Okay, it's not that it's very difficult and it cannot only get in laboratory. That is not the case. Question is, why we will read supercooled water? Water, we know, is a very peculiar liquid, and it has many anomalous behavior. So more than 50 anomalous behavior are known. And question is, sometimes we think that water is so peculiar, and how to understand water? To understand, this peculiarity of water increases as we reach the supercooled state. So to understand water, one has to understand the supercooled state. Then you can clearly understand the origin of this different anomalous behavior. Few anomalous behavior are here are summarized. There's 4 thermodynamic properties. You can read this article, this review article in Chemical Review, published in 2016, very beautifully expressed and shown. So this is like density. You can see, we know from our school days that density of water goes maximum at 4 degrees Celsius. while simple liquid with decreasing temperature, the density should increase, but for water, it goes through maximum and then again decreases. If you extend it, it is possible to see that density will again increase up to below a certain temperature. So, what is the reason behind it? Thermal expansion coefficient alpha. This is basically del V del P, 1 by V del V del P. So that is the coefficient. How with increasing pressure, volume changes. So here you can see that for normal liquid, as you decrease the temperature, this thermal expansion coefficient decreases systematically, but it does not go below 0 degrees. But what happens in water? It doesn't go below 0 for alpha. So it is always positive. So with increasing pressure, volume should increase. So this is thermal expansion coefficient. So with decreasing temperature, volume should decrease. But for water, what happens is below 0 degrees Celsius, it happens opposite. Not 0 degree, 4 degrees Celsius, it happens opposite. The isothermal thermal expansion coefficient goes negative value. That means with decreasing temperature, volume increases. That is interesting. And similarly, isothermal compressibility. And you can see above 46 degrees Celsius, it behaves almost like simple liquids. But below 46 degree, it starts increasing and almost shows divergent behavior. Same thing for heat capacity of water. The thing is, these thermodynamic quantities can be beautifully explained, this anomalous behavior, by their fluctuations of volume and entropies. For example, the isothermal compressibility is expressed by the fluctuations of volume, and heat capacity by the fluctuations of entropy, and thermal expansion coefficients is by the correlated volume and entropy fluctuations. And you can see here, when something is increasing, that means the fluctuations are increasing. So something is happening as we decreasing the temperature below a certain point, that the fluctuation is increasing, which should not be the case for simple liquid, because with decreasing temperature, fluctuations should decrease. So there are different scenarios which qualitatively and quantitatively explains this thermodynamical behavior. The first one was the stability limit conjecture, which was given by Speedy in 1982 in this paper. You can see here, it says that this is the spinodal, liquid vapor spinodal. So that is the maximum stability line of instability. So this is the critical point of this is the liquid water phase diagram. It's the critical point. It's decreasing and it goes negative pressures. And then when it touches this is the line of density maximum for water. And when it touches this line of density maximum, it changes its behavior and goes to the positive pressures. And as this goes to the positive pressures, then what happens is this line of instability is the reason for these fluctuations. That is what was proposed. But later on, in 1992, Stanley's group has discovered that when in this line, I will go into that next slide. It's not the line of instability, but this is a phase transitions line. Phase transition line between liquid-liquid phase transitions, and it ends at a point that is called critical point, because here also there is a critical point, liquid vapor critical point, and this is a liquid critical point. And two sides of this line are two different liquids, and once this point at the end of the point, there will be a line, hypothetical line, that is a Widom line, which will start, and that is and this line is basically the first order phase transition, liquid first order phase transition. So whenever you cross this line, there will be a fluctuation will increase. There are other approaches as well, critical point free scenario and singularity free approach. This scenario says that the critical point will be at this point where the liquid vapor spinodal and it meets with the load with the line of density maximum. That is the negative pressure. However, the second critical point hypothesis actually is more popular now, and I will explain you now. I will answer the question of Rajorshida. the detailed phase diagram of water, which actually emerges from this critical point hypothesis, liquid-liquid critical point hypothesis. This is the phase diagram, and you notice you go through this line vertically downwards, okay? And this is the pressure, zero pressure here. So very close to 1 atmosphere. Now, as you decrease the temperature, you first encounter the point written 0. That is the melting temperature at atmospheric pressure, above which it was stable water, stable liquid water, below which your thermodynamically ice is more stable, but liquid water is better stable, and that is why it is called supercooled water. But you cannot reduce the temperature as much you want. There will be a temperature 235 Kelvin at atmospheric pressure, where liquid water will instantaneously freeze. And that is why it is called homogeneous nucleation temperature, TH. So above which water will also freeze if you have a surface or some other impurities. What happens is that is called heterogeneous nucleation. But without any external objects or surface or something like that, impurities, water will instantaneously freeze also when it is pure, ultra pure state. Absolutely no impurities. And that temperature is called homogeneous nucleation temperature. Now you decrease further. It is not possible to keep water liquid, and it will be instantaneously freezing. If you remember the free energy diagram I have shown in the second slide, that minimum is actually going up so much that there is absolutely no minimum. So it will be spontaneously, it will go to the ice state. Now we do one thing, we quickly quench it very fast, and you will reach this glassy state. What is the glassy state for students? Glassy states are the structure like liquid water, dynamics like solid. Very slow, and glass means the structural relaxation time is more than 1 second, and the viscosity is more than 10 to the power 13 poison. Physical chemistry, in physical chemistry, we then call it as a glass. So that temperature, where it reaches that phase, that is called the glass transition temperature. For water, at 1 atmosphere pressure, this is 136 Kelvin. So if you quench the liquid water very, very fast, then you can reach the glassy state. Then do what? Then you increase the temperature again slowly. Then once you cross this glassy state, you can see my laser pointer. At these regions, you will again get the water liquid state. And that water is ultra viscous and is called deeply supercooled water. And it can continue up to 160 Kelvin. Everything, this number are all atmospheric pressure. So 160 Kelvin, you will encounter again another freezing. So you are heating it up, it will start again freezing. This is TX. So therefore, between TH and TX, you will not get water in the liquid state, but it will spontaneously freeze. And that is why these people, led by Stanley's group, they called it no man's land region, quote unquote. No man's land. So here you cannot get the water in liquid state. And experimentally also, it is still not been possible to get stable liquid state below this TH and above TX. Okay. So now what happens is, if you go to this glassy state, if you are in the glassy state, you increase the pressure, 2 different phases are there. Low density amorphous ice, high density amorphous ice. You can convert the low density amorphous ice to high density amorphous ice by increasing pressure. And this line was given by Mishima et al. In 1998 in their Nature paper. And you can cross this and you can reach the other region, and there is also a first order phase transition. And now if you extend this line, you will go in this region. Although experimentally it is not observed in liquid state, but theoretically you can say that liquid state can be present. It is possible. If you, in simulations, what happens is your cooling rate is infinitely fast. So, and your simulation time is very, very small, very short compared to the realistic time scales. So we can study water liquid in this no man's land region as well. And you have this line which separates these two different liquids, low density liquids, high density liquid. And it also ends at a point that is called the critical point. Second liquid critical point. Why second? Because already one critical point is there in liquid vapor, and this is the second critical point. And this is not liquid vapor critical point, this is liquid critical point, because either side of this line are liquids. So then what happens is there is a line emanates from this critical point, dashed line, that is the Widom line. The Widom line is the extension of the critical point. That is also there in the liquid vapor state. And the same analogy, this line was named as Widom line. Now what happens as you decrease the temperature at a constant pressure? First you are in the stable state, you are going to the supercooled state, then you are slowly crossing this Widom line. As you cross the Widom line, you are going from one liquid phase to another liquid phase. And around that point, there is a first order phase transitions. And we know when the first order phase transition is there, then there is a fluctuation. And fluctuation increases. Because the fluctuation increases, this different thermodynamic properties also increases. That is the explanation for thermodynamical.
So, Snehasis, why the Widom line is drawn with a dotted line?
Because this is the hypothetical line. There is not a physical line present. This is that is why this is dashed line. But like in the liquid vapor critical point, after the liquid vapor critical point, we think that supercritical region is like a homogeneous phase. But that is not the case. One side of the critical line, one of the critical point is like liquid, another side is like.
Got it. It is unlike the first critical point.
Yes.
Unlike the first critical point. So you don't have a second order phase transition there.
No. First order phase transitions,
but the line is not experimentally determined so far, but it is. Can you construct?
Because experimentally is difficult. It is in glassy state. It has been done.
Yeah.
Critical point is still debated and still doubtful, because experimentally you see the problem is the critical point is 500 bar to 223 Kelvin below the homogeneous nucleation. So, but people are trying very hard.
No. If you cross that the gray color line, this is the first order phase transition.
Yes.
But then you have a critical point. Ideally,
this is like one side of the dashed line is HDL line.
Yeah, it is just as if it is an extension of the gray line, but you have the critical point.
Yes.
But
so basically,
ideally, one should have a second order phase transition, like the usual critical point, but you don't have it here. Beyond that critical point,
most probably not. Acha, one thing here, what we should know, that critical point is not like a very isolated mountain. It's like Himalayan region. So in one talk, Stanley also explained that. Because what happens is in the critical point, you have an extension. And as you go far and far away from the critical point, your phase transition behavior will be diminished. Less and less you can see. So as you are close to the critical point, you will see is very, very intense change, intense fluctuation. But as you are away from this critical point, this thing will decrease. So slowly you will see a decreased impact on the fluctuation.
One more comment. Since you can distinguish between LDL and HDL, that's why you have that line.
Yes.
Otherwise, you would have not had that line.
Yes.
Okay.
So now thermodynamical anomalies were explained and it was studied so much in literature. You can see. But dynamical anomalies are less explored and less studied. Two thermodynamical anomalies are often discussed in literature. The most studied one is the fragile to strong transition. I will tell you what is this. And another one is the diffusion viscosity decoupling. So I should tell that we are interested in our group right now at this point, diffusion viscosity decoupling, which I will tell at the later stage. Let us first introduce what is fragile to strong transition. So you can see here Angel's paper, very beautiful paper. In 1999, he said that what is fragile and what is strong liquid? Fragile liquid is like, so basically, if you see the diffusion coefficients or any dynamical properties, it can be viscosity, it can be structure relaxation time, alpha relaxation time. they should behave like Arrhenius equations. You can see this is nothing but the Arrhenius equation. So D equals to D0 e to the power minus EA by RT. EA is the activation energy. So if you plot ln D versus 1 by T, you should get a straight line, completely linear. Now what happens is, some liquids are present which does not follow these Arrhenius equations. We call them fragile liquid. There's a name, fragile liquid. And some liquids are there which follows this Arrhenius behavior, and we call them strong liquid. So Angel in his paper in 1993 and 1999 particularly, said that water is in between these two, neither fragile nor strong. It changes its behavior as the temperature is changed. It is observed that at higher temperature or ambient conditions, the water does not follow the dynamics Arrhenius behavior. Neither diffusion nor viscosity nor alpha relaxation time, it does not follow. However, if you go and decrease the temperature, it starts changing. You can see the experiment done by Chen and Melamase in 2006 in their PNS paper. What they have shown is very interesting. At height, 3.3 means room temperature. So starting from room temperature, as you are decreasing the so you are basically decreasing the temperature, and you can see there is a non-linearity. But then below a certain temperature, that is 224 Kelvin, similar temperature of that critical, the Widom line temperature, it starts following Arrhenius immediately. Now you can ask, sir, how that experiment was done, because the temperature is very low. How the experiment was done? So the experiment was not done in bulk water. It was done in confined water. So water was basically confined in silica pores, and due to the confinement, the freezing point decreases, and one can see that you can see this is 200 Kelvin temperature without freezing. So this fragile to strong transitions was very interesting. This is the translational relaxation time, structural relaxation time. There also the similar behavior was seen in almost the same time. Now the question is, what are the explanation of them? Very nice explanation was given by this liquid-liquid critical point hypothesis. But the problem with that is, liquid-liquid critical point hypothesis says that there is a Widom line, and as you cross the Widom line, all the problems or all the fluctuation starts. But that does not explain the systematic behavior even at the ambient temperature. So this Tanaka's two-state model picture gave a nice explanation. It says, almost also in the same line of the critical point hypothesis, it says that water is not pure water. What does it mean? Water is not a pure state. It has two different liquids mixed all the time. It's not that you have to go to the near the Widom line, but even at ambient condition also there is a probability of having low density liquid. So there is a mixture. Water is basically a binary mixture of two different liquids. And as you decrease the temperature or you change the pressure, as you go along the phase diagrams, your composition, high density liquid and low density liquid changes.
So I have a question, Snehasis. So
yes.
So basically, you can can you go back to your previous slide? So basically, when you are at high temperature, you would need something like Vogel-Fulcher kind of plot, no, for this?
Now, Vogel-Fulcher-Taman equation actually fits at all temperatures.
That is
it
my
does
question.
Not show Arrhenius equations, but
but it will eventually be like Arrhenius equation beyond a temperature. When you
beyond a temperature, beyond that
below a temperature. But
yes.
What I'm saying is the transition from Arrhenius to Vogel-Fulcher, very sharp transition.
Yes.
So basically, your activation barrier is temperature independent at very low temperature, and then high temperature you have temperature dependence.
Yes.
One way of looking at it, I can see.
So now, what is the explanation? Very beautifully, I have somehow mistaken. I've forgotten to put the slides, which is recently published in 2016, I think, by Tanaka's group. they have shown it very nicely. They have changed the temperature from 500 Kelvin to 200 Kelvin, a very wide range of temperature. They have published a PNS paper. I should have shown that paper. Very nice explanations they have given. What is the explanations? Two different liquids are there. Actually, what happens is if you increase temperature at higher temperature.
Snehasis, I think somebody is asking. I just missed. Yeah, please go ahead. Who was just trying to ask one question?
Snehasis, just one question. So you mentioned that there are two types of liquids, right? One high density and the low density.
Yes.
So is there any experimental evidence or any proof for it?
I can't hear properly.
No. Is there? You mentioned that there are two types of liquids, right? Low density and high density. So is there a proof for that? Any experimental or?
So he's asking whether there is any experimental proof for low density liquid and high
Of course,
density
there
liquid.
is no, there is no direct proof as such, but there are different indications. I will give you one indications just now.
Okay.
For example, if you have a mixture, high density liquid and low density liquid, the good thing is that why this fragile liquid even at room temperature? The explanation is these two different liquids behave Arrhenius behavior. Very interesting. High density liquid separately behaves Arrhenius. Low density liquid separately behaves Arrhenius. Their activation energies are different. So when they mix together, they shows Arrhenius, non-Arrhenius behavior. Yes. At higher temperature, when you have a pure state, at very high temperature, low density liquid is almost negligible, zero. So that time it was seen that at that high temperature, it follows Arrhenius. At very low temperature, what happens is now high density liquid is very less. So low density liquid normally, there also it behaves Arrhenius behavior. So that is a very nice explanation that is how this paper.
So if I follow your statement, then if I even if I go to very what is that called? Very high temperature.
Yes, it follows
that part if I fit, that should fit Arrhenius.
Yes.
So Arrhenius here, but in between you have deviation.
In between, because these are binary mixtures and the compositions are all already.
So basically, your composition, the relative composition is a function of temperature.
Yeah, that is why that paper is very nice paper. I could have shown this.
Yes. So Snehasis,
I think So just 1 minute. I think let us just proceed with the talk.
Okay.
It will be easy for the student to go with the flow.
Okay, fine.
Yeah, so let us have the talk. Maybe we can have the interaction at the end. That would be better for the student.
Sure.
Yeah.
Okay. So now this paper actually came in 2018. It was a great article in the way that it also explains this Arrhenius, non-Arrhenius behavior, fragile and strong transitions of supercooled water. And they have shown rather than only just fragile to strong, they have two different transitions. Non-Arrhenius to weakly non-Arrhenius and strong to extremely strong. These two different transitions are there at two different temperatures, although they are very close to each other. And these crossovers are basically because of these two different states, changing of two different states, locally tetrahedral and distorted structures molecule between 220 and 230 Kelvin. Now, question is, this thing, apart from the fragile to strong transition, which is still a debated topic and not yet, you can say, validate, not yet explained properly. Our interest here is the diffusion viscosity decoupling, or in more general terms, Stokes-Einstein breakdown. So Stokes-Einstein relation actually says that this is a nice relation which connects these two quantities, diffusion and viscosity. Diffusion is a microscopic quantity which defines the self-diffusivity of the particles, single molecular diffusion, single molecular dynamics, and viscosity is a very macroscopic quantity. So these two quantities are linked through these equations called Stokes-Einstein relations, and you can see here d eta by t. This quantity is basically independent of temperature. It should be independent of temperature if the system, if the liquid follows Stokes-Einstein relation. But does it obeyed by water? Question is again no, it does not obey. Many different liquids, although this Stokes-Einstein relation have many approximations, and it's a very simple equations. Liquid is not so simple; they are very complex. They have different type of interactions, but still, surprisingly, many liquids, even many complex liquids, also follow the Stokes-Einstein relation to a certain degree. But water does not follow. Not only just water, even supercooled liquid, other supercooled liquid, glass formers, they also does not follow the Stokes-Einstein relation. So here, something does not follow means, what we are trying to tell is, if you plot theta by t, it should be constant and independent of temperature. If it is not independent, we say it is not followed. So that's what was observed in supercooled water in 2006 paper by the same group who have studied the fragile to strong transitions. And again, this system, this is not bulk water. Again, it is not a it is basically water inside cylindrical pores, silica nanochannels. You can see here it is not eta, but the translational and relaxation time, and this time actually normally is said to be proportional to viscosity. So you can say this quantity basically gives proxy about the viscosity. So it should be independent, but you can see as the temperature decreases below 240 Kelvin, it starts increasing, and there is a certain change of the slopes. But at this point, 225 Kelvin or so, but it does not follow. That is for sure. Question is why? If we see that phase diagram again, then there was a paper in just 1 year after that paper, and by Stanley's group. They tried to explain this Stokes-Einstein breakdown at different pressures. You can see that this is at low pressures, and pressure increases, and the Stokes-Einstein breakdown actually gets diminished. So you can say Stokes-Einstein relation is more valid at high pressure. Now, what happens is, if you can replace this temperature by T minus TW. TW is what? TW is that same Wiedemann line temperature. Then what happens? Because at different pressure, your Wiedemann line temperature is different, right? So now what happens as you replace the T by T minus TW, you can see all three different lines actually merge with one another. What does it mean? It means that this breakdown looks like it comes from the Wiedemann line. And that is the reason why at different pressures you can have different type of behavior. But question comes later when in 2015 paper, this Coppins et al. Actually published a paper, and they have seen a wide range of temperatures, and they have checked the validity of Stokes-Einstein relation direct. Previously, it was observed, but it was not direct validation. Why? Because firstly, it was not bulk water. Secondly, it was not the viscosity, but the translation and relaxation time. Although we know translation and relaxation time normally is proportional to viscosity, but sometimes it is observed that it is always not correct. Sometimes it is not proportional to the viscosity. So this was, you can say, one of the first direct proofs of the Stokes-Einstein breakdown, and very quantitative one. Although they could not go below homogeneous nucleus in temperature, very obvious, because it was not confined water, but a bulk water. Okay, so what is the interesting point here? Apart from their new experimental data, you can see the new results actually provided new insights. You can see previously it was thought that this breakdown, Stokes-Einstein breakdown, will start below 240 Kelvin or so, I say, but it is not the case. When you see quantitatively, the breakdown starts even at room temperature. You can see here. And the difference between other glass forming liquids is, for other glass formers, the breakdown starts into 1.3 into glass transition temperature. It is almost the general rule. For other glass forming liquids, the breakdown starts from 1.3 multiplied by Tg. But for water, it is 2.1 into Tg. So the breakdown starts much earlier. You do not have to go to supercooled state. Even at room temperature, ambient conditions, Stokes-Einstein relation is not followed, and it starts already breaking down. So what is the reason behind it? So while trying to explain it, we encountered another paper, and that paper actually was very nice in 2013. It showed that not only in supercooled state, you can see, very interesting, at this state, highly pressurized, superheated water, gigapascal range. And as you go along, you do not see any phase transitions from their phase diagram. No low density water, high density water is not present at this region. but you can see a severe breakdown of the Stokes-Einstein relation. So something is more fundamental. And this was stated that no, this liquid-liquid critical point hypothesis and two-state model does not seem to work. So what is the reason? So we try to understand this more from a very fundamental perspective and a very molecular level. So in this paper, you can see this Goldstein hypothesis. What it says is very interesting. Okay, sorry. So it says that there are free energy landscape, and they are very rugged one. When the particles move at a very high temperature, you are above these different hills and hills. So it is simple Brownian motion. It does not see any changes of the free energy while moving. But as you decrease the temperature, you will encounter particular temperatures. Then it cannot simply move via free Brownian motion. But the motions will be like moving from one cage to another cage via a barrier crossing process, via multiple barrier crossing process. So it cannot be simple Brownian motion. And previous articles, many articles, qualitatively told that there are different jump effects which are responsible for these different anomalous properties of dynamics. And these jumps are basically the faster diffusions at particular points, and that actually gives rise to this heterogeneity of the medium. At some points, molecules move very fast. At some points, molecules move very slow. And this dynamic heterogeneity is also responsible for the Stokes-Einstein breakdown, and it was observed for other glass-forming liquid in many works. But question is, explanations were more qualitative. How we can explain these things even more quantitatively? For that, what we have to do? We have to calculate the jump contributed diffusion. And how can we do that? So first thing is, you have to analyze trajectory by trajectory for each molecule. And you have to identify which are the jump effects. What are the jumps? For that, you have to define the jumps. Once you can characterize and define and count the jumps, you can exclude them from your raw trajectory. You can exclude them. Once you can exclude them, and then you calculate the diffusions from the remaining trajectories, and you will get a diffusion. So now you check if jump is the responsible, then these trajectories which are devoid of jump, already we have excluded the jumps. Those trajectories should give you the diffusions which should follow the Stokes-Einstein relation. Is it the case? That's what we try to investigate. So our method, which actually gives rise to this protocol, I will explain slowly. I'll explain you in briefly. 4 different methods, 4 different points are there. Firstly, you have to. So let us say this is the trajectory of a single water molecules, and it goes through small, through very rattle motions where it is moving in the same space almost, and also then is a large jump, and then again rattling and large jumps like that. So these are small diffusions and these are large diffusions basically. So first we will basically segment. We will make different segments of this trajectory in a particular time. What time we will choose? It can be arbitrary, but we have taken the this time. This time is basically comes from the non-Gaussian parameter. Non-Gaussian parameter is basically says what time the dynamics of the molecules at different directions are maximally unequal. So that means the time where the dynamic heterogeneity is maximum. That time T star we have chosen for this segmentation of the trajectories. First step, and then first segment, second segment, third segment, you choose different segments, and then what you do is you calculate the so basically this one small segment is nothing but like a polymer, right? And in polymer you have monomer lipids, and here the monomers have the different time points. So once you have that, you can calculate the radius of gyration. Radius of gyration is often used in polymer, protein, etc. So once the radius of gyration is calculated, you can have an effective displacement in a trajectory. And then you can multiply it, then you can get, so radius of gyration is that this thing, and then you double it, you will get the effective displacement at a particular trajectory segment. Now question is, you get all these different trajectory segments and they are effective displacements. Now what will you do with that? you have to characterize these different segments, and you have to define which one are the jump, so-called jump, and which are non-jump. How will you do that? Here, Dynamic correlations is basically the set here. Why? Because Dynamic correlation says that the displacement, Dynamic correlation gives you the displacement distributions. And normally, displacement distribution should be Gaussian if it follows normal homogeneous dynamics. But what happens is, in a complex liquid and also a non-uniform liquids, heterogeneous liquids, it does not follow Gaussian. You can see here, this is the Dynamic correlation, basically the displacement distribution at time t star. So 0 time and then t star time, what is the displacement? You get all the different displacement, calculate the distributions. And then you compare with the Gaussian equation. And you can see here, normally what happens is the simulated Dynamic correlations have two different crossing points. The crossing point at the far distance, beyond which you can see that there is a probability of having larger displacement. We call these segments where the effective displacement is more than this R2, it's called jump. Now let us see with this definitions, and once we define the jump, then and you can see this paper. These are the original papers from where we have taken this idea. So once the jump trajectory segments are there, you can count them, and if you count, you can get the frequency, jump frequency, and in each trajectory segments you calculate the average, how much it moves, and from these equations which comes from the random walk problem, the diffusion coefficients can be written as 1/6 jump frequency into lambda jump square. So you have the jump frequency, you have the average jump length square, average square jump length, then you can calculate jump. Fine. Now what? You have, so basically now we started hypothesizing that actually in this overall dynamic, we have two different diffusions. One is jumping diffusions, where the particles are moving very fast than expected. And then another one is the remaining one. What is the remaining one? We call it a residual diffusion. And what is the physical meaning of it? It is very simple, which are non-jumping. So basically small step displacements, which follows more or less grounding motion. So what we do is, so to prove that we can write this d equals to d jump plus d race, what we have done is we have used this cut and stitch model. What is cut and stitch model?
Yeah, your voice is breaking.
Voice is breaking?
I can hear.
Okay.
I did not have any problem.
Should I continue, Rajiv?
I can hear.
Okay, you can hear. Okay, I assume it is okay now. So
Yeah, it is audible.
Snehasis, are you
okay.
there?
Yes, it is audible properly.
Okay, fine.
Good. Yeah.
So now this method is cut and stitch method, just to prove this equation that diffusion is the summation of
Might
jump
be having
diffusion.
some network issue.
What we do for that, we cut the jump trajectory segments and stitch them.
Let us just
Cut the small diffusive trajectory segments, non-jumping segments, and stitch them. So what we will have effectively jumping trajectory, only jumping trajectory, and only non-jumping. Now, with the jumping trajectory, you calculate the diffusions via the mean square displacements. Non-jumping trajectory, you calculate the diffusions. You get the residual diffusions. You check whether this equation is valid or not. Unfortunately, this equation is valid, and we get the residual diffusions very easily by subtracting the jump diffusions for the total diffusions. So now, these residual diffusions, once we obtain, and we have published a series of papers on this topic. If you are interested, you can go through them. And these are the visual proof that, yeah, we can actually see the jumping segments and non-jumping segments. At the same time, you can see it has so much spreading, and here it is very small. Similar thing at two different temperatures. So now we calculate the residual diffusions into viscosity by T and the total diffusions. While this was like very high at low temperature, 210 Kelvin. And by the way, it follows the experimental trend very well, almost quantitatively, fully quantitatively. But the residual diffusions, when you calculated d raised eta by T, you see the red line that they are almost constant. You cannot say it is fully constant because there are error bars. In the error bar limit, you can say It is almost constant and almost at one. What does it mean? It means the residual diffusion part is basically following the Stokes-Einstein relation very well. And we have that leverage to get how much percentage of jump diffusions we are getting as the temperature is decreasing. It is very interesting. When we see the jump trajectory segments, means how many, so let's say you have 100 different trajectory segments, how many are jump trajectory? Only 5% out of 100. So 95% are non-jumping. Only 5% is jumping. But these 5% jumping segments actually can contribute almost half of the total diffusion. That is very interesting. This is the jump contribution. So basically d jump by d into 100. So that is the jump diffusion contribution in percentage. So at 200 Kelvin, when the percentage of jump trajectory segment only 5% is almost negligible, but it is dominating the diffusion by 50%. So that is what they deal. So with that success, we have also checked the same paper, Stelly's article in 2007. You can see that at different pressures, and when they replaced by T by T minus TW, all the different pressure lines merged with one another. Very nice. But we have done not only that they have merged, they are all merged around one. So not only that, these near rhythm line, all these stars and all these things, but also the actual fundamental origin of this breakdown is basically because of these overwhelming contributions, increasing overwhelming contributions of jump diffusions with decreasing temperature. So different structural properties we calculated to correlate why this thing is happening. I should mention this thing that the limitations of our work, still it is incomplete. Why? Because we still could not, we have not been able to correlate this. Why? Now, question is, three things are there. One is this two-state picture. I think, okay. So we have two-state pictures, high density liquid, low density liquid, we already explained. Stokes-Einstein breakdown. So what is the connection? We have connected a half part. That is the jump is the reason for this breakdown. But what is the connection between jump and this two-state model? Still, it is not clear. So we are going to do that. Most probably within 6 months or so, we will be able to successfully answer this question. Which part of the liquid is basically contributing to the jump trajectory? And which part? Okay, so that is what is remaining. Now, this translational jump diffusion approach is not only applicable for understanding this breakdown of Stokes-Einstein relation supercooled. Rajiv, how much time I have left?
So it's almost an hour. You can take maybe 5 or 10 minutes. 10,
15 minutes, okay? Because in the meantime, I was asked questions.
Try to make it quick.
Okay. So, so we can apply the translational jump diffusion approach in other fields. Particularly, I will talk on this topic, and if possible, I will also touch this. Okay, so anomalous dynamics of lipid membrane. What is this? So actually, there was an article in 2016 in Nature Communications that is an experimental paper. It said that there are dynamic heterogeneity and non-Gaussian statistics for acetylcholine receptors on live cell membrane. So cell membrane is there, and you have the receptors, which is basically protein, and these transmembrane proteins. And these proteins, when they move, they are moving via dynamic. The dynamics are heterogeneous, and not only the dynamics are heterogeneous, their statistics or dynamics is, although the diffusion is linear, you can see that diffusion is means per displacement is linear, but the dynamics, that the distribution of diffusion is non-Gaussian, highly non-Gaussian. What is the reason? So, and it is called Fickian means per displacement, but non-Gaussian diffusion, which is very rare. Now, question is, they have explained this using this Picket fence model. What is that? It says that this protein, when they move, lipid membrane is also lipid bilayer. I should tell to students, lipid bilayers are made of lipid molecules. They are basically self-assembled structures, and lipid have the head groups, and which are polar, and tails are the hydrophobic. So these tails actually comes closer to each other, away from water, and forms this hydrophobic core region, and hydrophobic groups actually face water and interact. So this is basically creates a barrier. Now, proteins actually also are anchored in this membrane and they move via diffusion motions. What is the nature of diffusion? It's very complicated. Okay, because a lipid membrane, cell membrane is also very complicated. Now, what they are explaining, why this dynamics is like that, what they are telling is because they are anchored with this actin network, which is beneath the lipid membrane or cell membrane. And because of this remodeling of the actin network, the dynamics is highly non-Gaussian. Very complicated. Okay, very complicated biology. And you can see the dynamics, the trajectory is very much heterogeneous. So we asked, really, is it required to explain this non-Gaussian, Fickian yet non-Gaussian dynamics using this complicated dynamic Pickett-Pence model, or it is even easily understood? Rajar Siddha is there, and he is very expert in this area. So we discussed a lot about this, and he also had a good inputs that you can see more simply about the dynamics of lipids, and we followed that. What we try to understand is, if we have, let's say, we know that one of the best popular model of cell membrane is the wrapped model, wrapped membrane. What is wrapped membrane? You have three different type of lipids, saturated lipids. You can see the blue ones. This is the saturated lipids. Saturated lipids means their tails are saturated. Unsaturated lipids and cholesterol, so they are there. Now, if you have a particular compositions, you can have in simulation also, you can have a very much heterogeneous structure, and you have a phase separation, liquid phase separations. These blue ones with the yellow, I think yellow color or green color, I don't know. They are basically liquid ordered phase, and these are liquid disordered phase, and they are ordered and disordered. These two different phase separations are there, and the dynamics here is very interesting. So what we have done a Martini simulations, a long simulations we needed because to see the dynamics. Dynamics is not very accurate here, of course, we know that. But the main problem is to get a proper wrapped structure, you need to have a hugely large system. And the time scale also very large. Okay, all atom simulations cannot do that. Or even if it can do, it will be a very huge task. Okay, so we did it with coarse-grained modeling, although we did not want that because we know the dynamics in the coarse-grained model is not good, because this work is basically the dynamics, but still we did not have any other choice at that point. So we saw that this wrapped structure is there, and now let us see what kind of dynamics of lipids we are getting, not proteins, lipids. Okay, you can see here these different lipids. DBBC actually is in the yellow phase, liquid ordered phase, DBBC and cholesterol. And DIPC, that is the unsaturated lipid, is with the liquid disordered phase. And you can see here at long time, the mean square displacements are all very much linear. Okay, and you can see the gamma, that is basically the differential of the MSD with time. They goes one, that means they are diffusing. Good news. But the problem is when you see the displacement distributions by Van Hoek correlations, you can see this highly non-Gaussian behavior. And this non-Gaussian behavior is not uniform. It is only happening in the liquid ordered phase, but the saturated liquid is there, not in the liquid disordered phase. Okay, so if the protein, or the hypothesis, if the protein is anchored in the liquid ordered phase, if the lipid also moves in this kind of dynamics, protein also is supposed to move in the similar dynamics. That is what they are theory. And you can see here that this Van Hoek correlation is basically exponential at a long time. It should be Gaussian, but it is basically exponential. And this correlation length is basically proportional to the delta T. Now, question is, how will you explain it? There are already known explanations for this Pickian mean square displacement yet non-Gaussian diffusion. One is a diffusing diffusion picture. Another is already very popular. That is continuous random walk. Okay, so these two pictures can explain, but we have also seen that we have explained that they are the reasons for which this kind of dynamics is feasible, possible. So side by side, we have tried to zoom or try to resolve more. So we have seen that this is the blue liquid ordered region. And if you can somehow separate this interfacial region and the core region, and you can study the same thing, you can clearly see that core region there is no non-Gaussianity, or non-Gaussianity is almost negligible. But in the interfacial region, the non-Gaussianity is very much vibrant. You can see here. And that is therefore we can say that this is not the characteristic of yellow region, but this is the characteristic of the interfacial region. Okay. And also another interesting thing is the dynamics of the molecules in the interfacial region, highly jumping dynamics. And that's what we try to study using our translational jump-diffusion approach. And we try to explain. You see, what most important thing, as it is non-Gaussian, and you can see here that the original displacement distribution is elongated at longer distance. That means there are large displacements possible for these systems, for these lipids. And that is exactly what we see, that the jumping contributions using the same translational jump-diffusion approach, we calculated how much percentage the diffusion is contributed by the jump diffusion. So you can see almost 20%, 16 to 20% at small time. But as we go along, it should decrease. But it's still not, up to 20 microseconds. What is 20 microseconds? You can see 20 microseconds is like where it is fully linear. There also, and also, even at 20 microseconds, there is a significant contribution of the jump diffusions which is coming from it. This is interesting. Therefore, long time persistence of jump diffusion contributions can give rise to this Picarian non-Gaussian dynamics. If you see these two theories, it falls in this continuous time random walk problem. Another applications, quickly I will tell, that is the now we can summarize what we have done. That if we have this, if we have the system, we can calculate the two different diffusions, jump diffusions and residual diffusions. We can separate them, and we can check the residual diffusions whether it is the Stokes-Einstein relation is followed or not. Now, if you put these things upside down, what happens is we have the diffusion coefficients in some situations. You can easily calculate diffusions, but you cannot easily calculate viscosity. Now, if you think about it, if we have the diffusions and if we can calculate the translational jump diffusion approach, and we can calculate the jump diffusion coefficient, and then subtract from the total diffusions, we get the residual diffusions, and then we use the Stokes-Einstein relation, I can get the viscosity. Very easy. So that is what exactly we have done in this approach. We call it. So why the problem actually is coming? You see, this is nanotube, water inside nanotube. And 1010 carbon nanotube is a very narrow 1.34 nanometers. And previously, there was a simulation study by this group, and they have shown that viscosity of confined water is 100 times smaller than that of bulk water, which is extremely surprising. Although diffusion is similar. So what is the reason behind it? Diffusion is similar, but viscosity is 100 times smaller. Is it the case, or some other problems? So unfortunately, they use this green correlation. And the green correlation, the problem is your system is confined in all sides. Only one side is open. That is z directions. If you consider z direction is the channel's axis. So when you have the pressure tensor terms from the green correlation, then the problem is pressure tensor terms in all directions are asymmetrical. And with this asymmetrical pressure tensor, can you really average it and get the viscosity? That is the question. So what is the so it is possible that this Stokes-Einstein relation is not followed. And then what is the another way to do? That is the Stokes-Einstein relation. Now you see the problem is this paper of Shubham and Professor Bagchi in 2022, they have shown nicely that for the confined system, this is basically a slit pore. So this is basically slab geometry. Two sides are open, so you can still use the green correlation very well. And they have used the green correlations to get the viscosity and diffusion coefficients parallel to the slab. And you can see very nicely as the distance between the two slabs decreases, the Stokes-Einstein relation is not followed. Both stick and slip hydrodynamic prediction fails for narrow slits. So then what will you do? You do not have a choice to use the Stokes-Einstein. That is what we have proposed. JCAC, jump confined, jump corrected confined Stokes-Einstein relation. That says that, as I said already, that if you have that diffusion coefficients and you have the translational jump diffusion approach, at least for one directions, you can calculate the jump diffusion coefficients. And in the confined Stokes-Einstein equation, you simply replace the diffusions by the residual. Already you have discussed about that. And then you calculate the viscosity, just upside down. And then what will happen? Stokes-Einstein relation is followed. Already we have shown that the residual diffusion is actually following Stokes-Einstein, and already confinement effect already taken into account. So then we have used this, and we have seen very interesting that as these are very different temperatures, we have reached and 3 different carbon nanotubes with increasing diameter. And you see where the narrower carbon nanotube, the viscosity is basically, I will go to the viscosity, that is the most important thing. Viscosity is much higher compared to the water inside compared to bulk water, and also compared to the water inside more broader, wider carbon nanotube. And you can see viscosity actually decreases. Wider carbon nanotube less than bulk water, which is also very interesting. And it was already observed previously that dynamics inside the carbon nanotube is faster until if the diameter for a certain diameters, and that is also correct. And also these trends actually follows the diffusion trends, and you can also explain that why the viscosity is so high inside the carbon nanotube of narrower diameter, because the hydrogen bonds are very strong. Although hydrogen bonds are reduced, but the particularly the tangential hydrogen bonds on the surface, they are very strong, and the hydrogen bonds are increasing. The free energy for breaking the hydrogen bond is increasing, and therefore the viscosity of the water is supposed to be higher, and which is consistent with the diffusion. So then we also try to check if we replace with superhydrophobic carbon nanotube, although it is not a real superhydrophobic carbon nanotube, we have just tuned the Lennard-Jones parameter to get the contact angle correct. Then you can have like superhydrophobic carbon nanotube is basically much less viscous compared to water inside superhydrophobic nanotube, much less viscous compared to bulk water, compared to hydrophobic and also bulk. That is very interesting story, and because people want to know how fast the dynamics of carbon water inside nanotubes or nanochannels, and it's a good story that for superhydrophobic, if you use superhydrophobic channels, then it will be less viscous. We have also tried to explain different non-monotonic behavior of water alcohol mixtures, and I will skip this thing. And so the thing is, what are the future directions? So in future we can use the as I already said, that already the connections between the Stokes-Einstein breakdown and the translational jump-diffusion approach is set. But the other part, which is the connections between the translational jump-diffusion approach and the existing theories of supercooled water, is still due. And we have already started the work, and hopefully we will come up with new solutions. And that why the jumps are happening, at what point the jumps are happening. Is it that another interesting thing I will tell, that as you see the percentage of jump diffusions, as it goes to the Widom line temperature, which is around 220 Kelvin, our jump diffusion contribution goes to 50%. And if you take the two-state model of water, there also you have a composition high density liquid, low density liquid 50-50 at the Widom line point. So is it just coincidence or some connections is there? We are going to analyze. And this kind of formalism you can also use to calculate the viscosity, and also it is useful for elucidating dynamics in other biological systems. And I acknowledge my students, Vikas and Shivam. They actually started working on the supercooled water. Sakira worked on this lipid membranes, the Fickian and non-Gaussian dynamics. Golam is working on the water inside nanochannels and different funding agencies. And you can see this is Sakira. She is doing postdoc now. And Golam is carbon nanotube guy. Shivam started working on the supercooled water, and other students and funding agencies. And thank you all for your kind attention. You may please ask some questions.
So, thank you so much, Snehasis, for this wonderful talk. And we have many questions in the chat box, and I will go one by the other. So, very first question was asked by Abhilasha, and the question is, why there is barrier in free energy diagram what is the phase there at top of the barrier? I think when you were discussing the free energy profile, referring to that slide.
Very good.
In the very
Very
beginning.
good question. I could understand. So what is the explanation of the barrier? What is the origin? This is the entropic barrier. If you see, when supercooled water is there in one side and you have ice in another side. Sorry, I will move switch off the video in one computer. So one side you have liquid water and you have another side you have ice. So why the barrier is coming? Barrier is coming because of the entropy mostly. Why? Because when liquid water is basically entropically high, entropy is higher compared to the ice. So when it goes from liquid water to ice, the entropy decreases. Although enthalpy is gain, but entropy decreases. Still, the barrier is there because you have to nucleation. This is the process of nucleation, and this barrier is basically the barrier of nucleation. It has to cross, otherwise it will not go to the other side.
Yeah, and the second, a couple of questions were asked by Ravi Kishore. Yes, you can, if you open the chat box, you'll be able to see all the questions. So, Ravi Kishore,
and then what differentiates the green region from the yellow region?
That is regarding your phase diagram where you showed the
So green region is basically the stable liquid, ambient liquid below the above the 0 degrees Celsius, and I think that is the color, if I'm not wrong. Let me see. Okay, so I think this is green color, right? This is green color. A stable water is the green color, right? And supercooled water is the yellow color. I'm not very clear about green and yellow. I'm colorblind. Okay, so this stable water, that means above the melting point, freezing point, that is stable water, and supercooled water is that above homogeneous nucleation temperature to melting point. So, for example,
I think Snehasis, maybe he was asking, I don't know, maybe between ultra viscous versus supercooled.
Oh,
I think so. I think so.
green
Not,
color segment.
Got it.
Ultra, like deeply supercooled versus supercooled, yeah.
As I said, if you quickly quench it, you will go to the glassy state. Then again, you increase it, you will get liquid state, very tiny range. And that liquid is very much highly viscous liquid. So much viscous that it cannot also orient or arrange themselves to freeze, because for freezing, for getting ice, your water molecules rearrangement is required for getting ice. So that rearrangement time scale is also like much faster than the dynamics of the molecules. So then the problem is it remains in the liquid state. You can get it, and that is called deeply supercooled water. Hope that is what you were asking.
Yes.
Regarding the fluctuations, another question. So, are there density fluctuations? If yes, what is the length scale? Have this been seen in nano?
Yeah, that is a very good question. So this is a density fluctuations. You can say volume fluctuations, and also fluctuations of order, because high density liquid and low density liquid, these two different states are very different because if you see their structures, what is the structure I should tell? High density liquid is basically when you have the fifth water, which also comes into the tetrahedral network of the central water. So basically what happens is the effective volume is less, and also the density is higher. That is why it is called high density liquid. And when it is away from it, then we call it low density liquid. So therefore, volume fluctuations, of course, entropy fluctuations, order fluctuations, all those things. Both being supercooled water. Okay, sorry.
Okay. So, and there is another question, one more questions by Siddhant Parikh. What is the reason at the molecular level behind these jumps?
Very good.
And what is the molecule design so that it can be increased at some other temperature?
Very good.
So that overall diffusion will be increased.
So again, I will come to that slide. this Goldstein's hypothesis is very simple, yet very much elegant. Why? Because you see, Goldstein's hypothesis says if your temperature is high, so it's a hot liquid, then you do not have any barriers to cross for dynamics. So it will be simple Brownian motion, free Brownian motion. But as the temperature decreases, it's like you can see the seabed where you have a lot of hills and hills. So when you move through this, or any organisms move through this deep sea organisms move through this, it has to cross the hills and then go down to the other side. So it's no longer the simple Brownian motion, free Brownian motion. Rather, it will be this motion, diffusions will be like crossing from one barrier to another barrier. So that is why, so if you think if it is inside a local minima, it will simply try to escape the minima, right? Once it has a sufficient energy, it will cross. Once it has sufficient energy, it will cross to the other minima, and then again it will fall. So it will be like jump and caged motions, one after another. It will be there. And as the temperature decreases, so in all cases, it's not just about water. All liquids will have this kind of dynamic at low temperature, particularly. Okay, and if you see in the molecular level, then what happens is when the molecules are jumping, that is basically what is the physical picture of cage. Physical picture of cage is like your water molecules are clustered. Your water molecules are like trapped inside several other solvent molecules. Okay, and they are arrested. Now, once some other situation comes, or of course they are all thermal fluctuations. When it comes, the barrier opens up, and the particles go and sit in a new environment, new surroundings. So that is what you can say jump. And when the temperature is low, that the hydrogen bond interactions for water are strong. So the cage also is very strong. So it will open up, but it will take time. But when it opens up, it will have large motions, large dynamics. It's a hopping motions. It is also visible in solid states. In solid states also, ions move from one point to another point if you have defects. Very similar to that.
Okay. Thank you, Snehasis. So we have one more comment by Sridhi Parvi. Wrote that he's got confused because during the talk you mentioned that water is a mixture of LDL and HDL at high temperature. It is HDL, and low temperature it is LDL. If it is so, why isothermal compressibility shows singularity-like behavior? It should usually show a maxima.
So it's not that at high temperature only HDL is present and low temperature LDL is present. That is what the picture of Tanaka. Tanaka's two-state model hypothesis says that at all temperatures, all kind of state is present. Even at ambient temperature also you have low density liquid, but the composition varies. And at the Widom line, what happens is you have 50-50 compositions. Half of that are high density liquid, half of them are low density liquid, maximum fluctuation. And that is why maximum fluctuations give rise to the maximum value at that. So that is what the explanation. So it's, but in liquid critical point hypothesis, it says that it crosses through the Widom line. And once it crosses through the Widom line, it is a phase transition and it's like a singularity. But in reality, what happens is, and also if we do the simulations, you can see all water does not behave same. It is not possible that one side of the, even in liquid vapor critical point also the same story. One side of the line, hypothetical line, is liquid-like. Another side is vapor-like. Although it is said it is homogeneous liquid, it is not.
Okay, I think Vimanda has questions. So Vimanda, can you please unmute and go ahead?
Yeah, just one clarification I would like to have here.
Yes.
Snehasis, about your dissecting the trajectory into different kind of time T-star, right? That's the interval you have taken.
Yes.
And that T-star you are taking from the non-Gaussian parameter, right?
Yes.
Yeah, so if I understand, that this non-Gaussian parameter and setting in of diffusion in your linear MSD in your system is correlated. That means what I'm trying to say that this is, if this is your alpha 2t plot, and if you really calculate the MSD in your system,
yes, then it is just the onset
then you
of
will see, let me finish, then you will see till T star, it will be subdiffusive.
Yes.
And then only the diffusion will set in. Like linearity will set in. Linearity with T.
Yes.
It
So
is well
actually what
known
happens
in
is,
glass literature. You can have a check.
yes, yes. So T
Now,
star will be the onset of the linearity, you can say. Onset
yes,
of the
T
linearity.
star is the onset of linearity. That means linearity will be above T star, not below.
About this, no, you can say the crossover, where the substitution and the linearity
That's my point.
at that point. Exactly. Yes,
So
exactly.
now the point I'm little bit confused here. If you have your trajectory length, which is equivalent to your T star, then how come from there the diffusion you are getting from D rays is actually giving you the real density which can be scaled with Stokes-Einstein relation?
So, okay. So your question is like, your T star, you are taking T star as the time for the segmenting the trajectory, right?
Yes.
Now if you compare the mean square displacement and T star, your linearity just starts at T star. Okay.
Yes.
The point is why we have chosen the T star.
Yeah, I mean, no. Why you have chosen T star?
Right?
That's a different question. But I am telling that if you take T star, then whatever the data you are getting in a particular interval,
yes,
there the diffusion or dynamics has not crossed to the full diffusion limit yet.
No, it is not crossed. Of course, it is not crossed. But it has just started.
Then from there, the D rays you are getting, how that is the pure diffusion you are getting, which should be scaled with Stokes-Einstein relation.
Residual diffusion, whatever we are getting, this is the remaining trajectories, right? Which are not taken into account in the jump events. So that is the remaining trajectory segments. So there you do not have. So when you have, let's say this is the, I should tell this go to the slide. So here, you see, at this points, you are saying this point to this point is my T star. Okay. So now, so what happens is what we do is normally we did that this point, actually we set this at this point. So as if in your trajectory, you do not have now this T star, you have a continuous trajectory.
So that means you stitch.
Yes, it is now continuous trajectory. Now you calculate the mean square displacement again. There you are not taking this T star for the mean square displacement to get the diffusion. There you see the linearity. So you are basically segment dividing these two trajectories. You are getting two trajectories. Exactly the point by point, phase space points, XYZ, and the time. You basically artificially keep them at that point. And then you get the whole trajectory, you calculate the mean square displacement, see where the linearity comes, then you calculate diffusion. So whenever residual diffusions we are calculating, it's not that at that T star we are calculating. It is basically the linearity.
Okay. So kind of, let's say I kind of understood a little bit from here, whatever you were telling. So now the point, just last one point, one clarification.
Yes.
That means you are just, the caging one, you are just taking it out.
Okay, no, not caging one, the jumping ones.
No, D rays is you are getting from the caged like trajectories only, right?
No, D rays is all these things. Just excluding the jump. Cage is just to show visually that you can see the cage are there. These are just visual proof.
Okay.
But ultimately,
I got
what
your point.
is residual diffusion is the whole thing.
Whole thing, caging as well as the next thing you are getting.
Normal one.
Only the long jumps are like
only excluded. Only the long jump, which is just 5% of the total trajectory.
Okay. I got it.
Yeah.
Okay. Thank you.
Okay, thank
Yeah.
you, Devanda.
Thank you, Vipanda. I think Devdas has a question, so can you go ahead,
Yeah.
Devdas?
Yeah, since this is the nice
Yes,
talk,
Devdas, please.
nice talk. I have a couple of questions. The first one is, when you raise like pressure, when you go to high pressure, we know that density fluctuations increases, right? And then
A high pressure means at which point you are in the phase diagram.
so this is, I'm talking about this Stokes-Einstein deviation of Stokes-Einstein.
Oh, okay.
You have shown that at high pressure, it is deviating lesser as compared to low pressure, right?
Yes.
The plot, yes. So what is the reason behind this? Because
Very simple, very simple. So what they have told, you see, you are increasing pressure. So you are encountering the Widom line at lower and lower temperature.
okay.
You can see the Widom line is bent, carved. So as you increase the pressure, you are encountering the Widom line point lower and lower temperature. So for example, if you are at 1 atmosphere pressure or 0 MPa, then whatever temperature you will encounter the Widom line, that will decrease significantly as you go to 200 megapascal, for example.
Okay.
Because the line is bent.
Yes.
Okay, so Widom line is basically your pressure and temperature both are changing. This is the one phase coexistence line.
Yeah.
So that is why you are getting this.
But based on the definitions, the probability of having both the HDL and LDL will be 50-50 along this
50-50 at the Widom line. Yes.
So even if you go to lower temperature, it will be 50-50.
Yes, that's what.
But
So even if you go to the higher pressure, they are also at the Widom line. You should get, or actually, in simulations, when you people does, they don't get it 50-50%. Literally. Means because they're very difficult to get the actual order parameter. What order parameter you will choose? So, but that is the hypothesis that at Widom line point, you should have 50% high density liquid, 50% low density liquid. But because the temperature is changing with increasing pressure, so that's why you're okay.
So
that's why you're
another question is on this lipid thing. This is just like I want to understand.
yes.
So when you said the diffusion of that lipid molecule, it's just the diffusion of center of mass of the lipid?
Yeah. So yeah, center of mass.
Center of mass of this
Yes.
lipid. Okay.
Yes.
So this is and then one question that is like I just want to get to know about your opinion of that. So if you go to the first or second slide, so the anomalies, the starting of the anomalies happens for different properties. It happens at different temperature, right?
Yes.
So especially, yes.
Yeah, you are right. You are right.
Yeah.
At different temperatures.
Do
Yes.
you have any comment on like, because if let's say supercooled in the supercooled state having
I
this
understand.
yeah.
I understand. I can say only that maybe because this, if it is because of the fluctuations of different thermodynamic variables, then I can say only that the fluctuations are different. For example, when you have alpha, then there is a different fluctuations. When you have kappa T, then there is a different quantity. So different quantity behaves differently. Their behavior with temperatures are different, and that is why it can be.
Oh, okay.
I do not have much better answer than this.
No, I think that is a good point actually, because this compressibility generally we track in experiment also, like
Yes,
we track, and that is related to density only, like volume of density, and then on the other alpha P has entropy also.
Yes, yes.
Okay. Thanks for the
Thank
clarification.
you, Victor. Thank you.
Okay, yeah. Dash, is there any more questions?
I have just one comment.
Yes, Rajesh.
Just comment is that I think the technique you used, it was I think introduced by or maybe used by this guy, Samios or someone.
You see, I
Raptis
have
and Samios.
participated. Rajoshida, you have
Yes.
not seen it.
Yeah. So I just missed that part actually. Yeah.
Wow, I definitely have played it.
Yeah.
And this paper,
Yeah.
this paper, you see, Raptis.
Yeah. This is the one. Yeah.
This paper, if you go to their citation, only I have cited.
No, we have also cited because we have written.
You have also cited, yes.
Yeah. Just a comment. I thought, okay.
But of course, the methodology will not be exactly same. I would tell that this Arak et al., that is basically the Margulis group, they modified it, and whatever the things we have, mostly motivated by that.
Thank you. Yeah.
So is there any other questions by any of the participants? You can raise hand. I think the last question was asked by Ravi Kishore. Could you also comment on rotational diffusion? That might be the last
Yeah, that is a very good point and interesting point. And we are trying to, for a long time, these what are rotational dynamics and rotational jump. Lage and Heinz have done beautiful works, but in these works, their methods of getting the jumps actually are limited to the hydrogen bonded liquid, I think, because the definition of the jumps are based on their hydrogen bond partner exchange. We wanted from from day one to get a very generalized picture of this rotational jump, and you can use that for in any molecule, any liquids which does not have hydrogen bond and non-protic solvents also. And how we wanted to do that, I can tell you, although we have not been able to do this, and we are not getting time, or our students are not that interested, but these are also very important problems. So for example, here you have this trajectory, right? The translational trajectory. You can convert it into you can think of it rotational trajectory similarly. In a your axis will be different. Instead of x, y, z, you will have theta and phi. With these two angles, you can draw a trajectory, and you can calculate the radius of gyrations and get the overall displacements without any hydrogen bonds point. And then we can do the similar thing, because rotational diffusions also with the similar counterparts of all, like translational, like Hanover correlations, alpha 2p, rotational jump diffusions. One can actually do this, and one can get the breakdown and finality of Stokes-Einstein equations, which actually follows the rotational, which actually gives the connections between rotational time and the viscosity. But unfortunately, we have not done yet. But it will be a good thing to do in near future.
Thank you. I don't see any other questions. If not, let us thank today's speaker for the wonderful talk, followed by a very detailed discussion session. So once again, from all the coordinators, I thank Snehasis for accepting our invitation and delivering.
Thank you, Rajiv, so much for conducting so nicely, and thank you all the coordinators and Professor Bakshi also to give me the chance. Thank you.
Okay, Snehasis, thank you, and thanks all for participating and actively asking questions and taking part in the discussion session. So till we come for the next event, we'll say bye. So see you in the next event. Take care.
Bye.
Bye, Snehasis.
Bye.
Bye.